The uvula, uvula vermis, uvular lobe, forms a considerable portion of the inferior vermis. It is separated on either side from the tonsil by the sulculus folliculae, at the bottom of which it is connected to the tonsil by a ridge of gray matter, indented on its surface by shallow furrows, and hence called the furrowed band. The tonsilla, tonsilla cerebelli, amygdaline nucleus, is a rounded mass situated in the hemispheres. Each lies in a deep fossa termed the bird's nest, nidus avis, between the uvula and the biventral lobule. The uvula and tonsillae form the lobus uvulae. The pyramid and biventral lobules constitute the lobus pyramidus. The pyramid is a conical portion forming the largest prominence of the inferior vermis. It is separated from the hemispheres by the sulcus folliculae, across which it is connected to the biventral lobule by an indistinct gray band, analogous to the furrowed band already described. The biventral lobule is triangular in shape, its apex points backwards, and is joined by the gray band to the pyramid. The lateral border is separated from the inferior semilunar lobule by the postpyramidal fissure. The base is directed forward and is on a line with the anterior border of the tonsil and is separated from the flocculus by the postnodular fissure. The tuber vermis, tuber valvulae, and the inferior semilunar lobule, lobulus semilunaris inferior, posterior superior lobule, collectively form the lobus tuberus, tuberae lobe. The tuber vermis, the most posterior division of the interior vermis, is of small size and laterally spreads out into the large inferior semilunar lobules, which comprise at least 2/3 of the inferior surface of the hemisphere. Internal structure of the cerebellum. The cerebellum consists of white and gray substance. White substance. If a sagittal section be made through either hemisphere, the interior will be found to consist of a central stem of white substance, in the middle of which is a gray mass, the dentate nucleus. From the surface of the central white stem, a series of plates is prolonged. These are covered with gray substance and form the laminae. In consequence of the main branches from the central stem dividing and subdividing, a characteristic appearance named the arbor vitae is presented. If the sagittal section be made through the middle of the vermis, it will be found that the central stem divides into a vertical and horizontal branch. The vertical branch passes upwards to the culmen monticuli, where it subdivides freely, one of its ramifications passing forward and upward to the central lobule. The horizontal branch passes backwards to the folium vermis, greatly diminished in size in consequence of having given off large secondary branches. One, from its upper surface, ascends to the clivus monticuli, the others descend and enter the lobes of the inferior vermis, that is, the tuber vermis, the pyramid, the uvula, and the nodule. The white substance of the cerebellum include 2 sets of nerve fibers. 1. Projection fibers. 2. Fibrae propriae. Projection fibers. The cerebellum is connected to other parts of the brain by 3 large bundles of projection fibers, that is, to the cerebrum by the superior peduncle, to the pons by the middle peduncle, and to the medulla oblongata by the inferior peduncles. The superior cerebellar peduncles, brachia conjunctiva, 2 in number, emerge from the upper and medial portions of the white substance of the hemispheres and are placed under cover of the upper part of the cerebellum. They are joined to each other across the middle line by the anterior medullary velum and can be followed upward as far as the inferior colliculi, under which they disappear. Below, they form the upper lateral boundaries of the fourth ventricle, but as they ascend, they converge on the dorsal aspect of the ventricle and thus assist in roofing it in. The fibers of the superior peduncle are mainly derived from the cells of the dentate nucleus of the cerebellum and emerge from the hilus of this nucleus. A few arise from the cells of the smaller gray nuclei in the cerebellar white substance, and others from the cells of the cerebellar cortex. They are continued upward beneath the corpora quadrigemina, and the fibers of the two peduncles undergo a complete decussation ventral to the sylvian aqueduct. Having crossed the middle line, they divide into ascending and descending groups of fibers, the former ending in the red nucleus, the thalamus, and the nucleus of the ocular motor nerve, while the descending fibers can be traced as far as the dorsal part of the pons. Cajal believes them to be continued into the anterior funiculus of the medulla spinalis. As already stated, the majority of the fibers of the ventral spinocerebellar fasciculus of the medulla spinalis pass to the cerebellum, which they reach by way of the superior peduncle. The middle cerebellar peduncles, brachia pontis, are composed entirely of centripetal fibers which arise from the cells of the nuclei pontis of the opposite side and end in the cerebellar cortex. The fibers are arranged in three fasciculi, superior, inferior, and deep. The superior fasciculus, the most superficial, is derived from the upper transverse fibers of the pons. It is directed backward and lateralward superficial to the other two fasciculi, and is distributed mainly to the lobules on the inferior surface of the cerebellar hemisphere and to the parts of the superior surface adjoining the posterior and lateral margins. The inferior fasciculus is formed by the lowest transverse fibers of the pons. It passes under cover of the superior fasciculus and continued downward and backward more or less parallel with it to be distributed to the folia on the undersurface close to the vermis. The deep fasciculus comprises most of the deep transverse fibers of the pons. It is at first covered by the superior and inferior fasciculi, but crosses obliquely and appears on the medial side of the superior, from which it receives a bundle. Its fibers spread out and pass to the upper anterior cerebellar folia. The fibers of this fasciculus cover those of the restiform body. The inferior cerebellar peduncles, restiform bodies, pass at first upward and lateralward, forming part of the lateral walls of the fourth ventricle, and then bend abruptly backwards to enter the cerebellum between the superior and middle peduncles. Each contains the following fasciculi. 1. The dorsal spinocerebellar fasciculus of the medulla spinalis, which ends mainly in the superior vermis. 2. Fibers from the gracillin cuneate nuclei of the same side and of the opposite sides. 3. Fibers from the opposite olivary nuclei. 4. Crossed and uncrossed fibers from the reticular formation of the medulla oblongata. 5. Vestibular fibers, derived partly from the vestibular division of the acoustic nerve and partly from the nuclei in which this division ends. These fibers occupy the medial segment of the inferior peduncle and divide into ascending and descending groups of fibers. The ascending fibers partly end in the roof nucleus on the opposite side of the cerebellum. 6. Cerebellobulbar fibers, which come from the opposite roof nucleus and probably from the dentate nucleus, and are said to end in the nucleus of Dieters and in the formatio reticularis of the medulla oblongata. 7. Some fibers from the ventral spinocerebellar fasciculus are said to join the dorsal spinocerebellar fasciculus. The anterior medullary velum. Velum medullare anterius, valve of Vincennes, superior medullary velum, is a thin, transparent lamina of white substance which stretches between the superior peduncle on the dorsal surface of its lower half. The folia and lingula are prolonged. It forms, together with the superior peduncle, the roof of the upper part of the fourth ventricle. It is narrow above, where it passes beneath the inferior colliculi, and broader below, where it is continuous with the white substance of the superior vermis. A slightly elevated ridge, the frenulum veli, descends upon its upper part from between the inferior colliculi, and on either side of this the trochlear nerve emerges. The posterior medullary velum, velum medullare posterius, inferior medullary velum, is a thin layer of white substance prolonged from the white center of the cerebellum above and on either side of the nodule. It forms a part of the roof of the fourth ventricle. Somewhat semilunar in shape, its convex edge is continuous with the white substance of the cerebellum, while its thin concave margin is apparently free.